bless your holy name in the name of Jesus. We pray as we are about to begin this webinar. We pray for directions, we pray for grace, we pray for mercy. We pray that give us insight into what we are about to learn in the name of Jesus, that at the end of it, knowledge shall be imparted unto the hearers to the glory of your holy name. We thank you in Jesus' name. Amen.
Thank you very much, Dr. Sari. And whenever you enter a house, you want to be sure if you are welcome or not. And we are fortunate today to have the Deputy Director General of the Ghana Atomic Energy Commission to welcome us, to tell us if we are welcome, whether it is it is right for us to have this discussion. So, with a round of applause, please welcome up front Professor Dixon Adumakong.
Thank you very much, MC, and good morning to you all, ladies and gentlemen. All existing protocol. Good morning to you all, ladies and gentlemen, all protocol observed. We are gathered here to listen to a panel discussion on the topic, the science of genome editing, principles and application. And I believe this is going to be a very interesting topic. And I believe the panel and those gathered here will be able to ask more questions to understand the new area that we are all heading towards. This forms part of Gaec at 60 celebration. This activity forms part of our 60th activity celebration. Yesterday, we were at the Lorry Park having a malaria day. And today we are here. And I believe at the end of the day, we will go home with a lot of ideas to be able to propagate the genome editing in our society. I wish you all the best and God bless us all. Thank you.
Thank you very much, Prof. I want to call on the director of Bnari to give us a background and an idea what we should expect after this webinar.
Thank you very much, MC. Deputy Director General, colleague directors, fellow scientists, our distinguished guest speaker, our colleagues online, our very loved farmers who are in our midst, media, ladies and gentlemen, all protocols observed. I want to welcome you to this morning's webinar, which forms part of Gaec 60, as the DDG has alluded to earlier. Bnari has been the leader in the area of modern biotechnology in this country. We shepherded all the processes that culminated in developing the policy around the implementation and practice of modern biotechnology in this country that resulted in the establishment of the National Biosafety Authority, which we still work closely with. We have our scientists who provide technical support to the National Biosafety Authority. We have also helped to build capacity in the area of biotechnology over the years. A lot of students pass through our laboratories to learn the basics of biotechnology. We have had scientists come to work in our laboratories. We have had people from outside the country trained in our laboratories in the area of biotechnology and modern tools in breeding. In the area of modern biotechnology, we are actively involved in all the aspects that involve the genetic engineering, and now we are into genome editing. And over the years, Bnari has built capacity in this area. And in the past recent years, we have had two of our colleagues go through rigorous training in genome editing, and they are leading our genome editing project here at the Biotechnology and Nuclear Agriculture Research Institute. Thanks to the African Orphan Crops Consortium and the Africa Plant Breeders Academy, they provided that training, and we have had Dr. Andrew Sarkodie appear, and then Professor Samuel Amite participate in this training. Now we are one of the only two institutions in the country that have the platform for practicing genome editing in the country. It's Bnari and Waki at the University of Ghana that have been certified by the NBA to practice genome editing. And we are fortunate to have an expert with us to support our efforts in genome editing. And this expert came as part of the efforts of the African Orphan Crops Consortium, helping us to build capacity in this area. So they sponsored his coming to Bnari to come and train our staff. So beyond the two who have received training, we have other people whom these two are training, and then the expert is coming to give us additional support. Now, with the establishment of the genome editing platform and the prospects that we have as far as genome editing is concerned, I see a bright future for agri-food systems in this country. Because the genome editing technology is a versatile technology that can help us do a lot of things that would help us overcome the challenges that we face in our agri-food systems. You can edit plants to be resilient to climate, to be resilient to biotic stresses like pests and diseases, to be more nutritious, to have higher yield, and whatever you can think of. So it's a very important technology that we are adopting as a country. And we are happy that Gaec, at our 60th year, we are beginning to enter into these new areas and starting new course in the modern applications of biotechnology. I would at this point want to say a big thank you to our numerous partners who have made this happen. And as I said earlier on, the African Orphan Crops Consortium has been very supportive in helping us build capacity. The African Plant Breeders Academy, the AUDA NEPAD have been very supportive of our efforts to develop the area of biotechnology. Our national partners, WACI, WACBIB, CSIR, and the National Biosafety Authority have all been very supportive, and we have representatives from all these places, all these people here with us this morning. I will not forget our farmers who we work closely with, and they are here to be part of this webinar. And we are sure that once they understand these technologies, when we develop genome edited crops, they will be willing to adopt these crops to improve their livelihood. And not to forget our friends from the media who have been very supportive in helping us to create awareness around the genome editing technology. So I will not want to go into the technicalities because we have very qualified experts here who will take us into the technicalities of genome editing. So I want to just welcome you and ask you to sit back and enjoy this morning's webinar. Thank you very much.
Thank you very much, Professor Sai. I'm not part of the. Yeah. So, in ending his remarks, Professor Syme mentioned that we are privileged to have experts amidst, and it will be right for us to know who these experts are. We are fortunate today to have Professor Valentine Otan Ntui, who is a professor of plant genome editing at the African Genome Center at the University Mohammed VI Polytechnic in Morocco, and also at the University of Calabar in Nigeria, where he teaches and supervises both undergraduates and postgraduate students. He obtained his PhD in plant biotechnology, where he specialized in genetic engineering and molecular biology from the Chiba University in Japan. Today, you realize that there are two Japanese and one Australian in our midst. Then he did um, the Japan Society for Promotion of Science postdoctoral fellowship at the same university he obtained his PhD from 2011 to 2013, and proceeded to do another postdoc at the Center of um, Desert Agriculture at the King Abdullah University of Science and Technology from 2014 to 2017. After that, he joined the IITA as a postdoc and subsequently as a scientist, where he worked till last year. His research focuses on the development of agricultural cropping or agricultural crops using molecular biology, genetic engineering, and genome editing tools. And he has extensive experience in plant tissue culture and genetic transformation, gene cloning and plasmid construction, and the most recent one, CRISPR-Cas mediated genome editing, among others. Um, he's a mentor to several graduate students, undergraduate students, doctoral students, and all. And he's a member of the editorial board of a lot of international journals. He himself is an accomplished publisher with over 70 publications in peer-reviewed journals. And he's given presentations in several conferences across the world. Currently and previously, he secured research grants from a lot of international and bodies across the world. Ladies and gentlemen, our keynote speaker, Professor Valentine Ntui. Can we give him a round of applause? Okay. And then, so you hear later from him. And we have two of our own. Let me introduce the host centers manager, and then I'll go to the host training institutions manager or head. Dr. Andrew Sarkodie-Appiah is a senior research scientist, and he's currently the manager of probably the youngest center in Gaec, Plant Disease Research Center. And he has over a decade of research experience in plant molecular virology, which has culminated in him publishing in several peer-reviewed journals. His major skills include, but are not limited to, DNA and RNA extraction, PCR, which is polymerase chain reaction, and the most recent one, quantitative real-time PCR, cloning, genetic transformation, enzyme-linked immunosorbent assay or ELISA, DNA sequencing, and analysis of sequence data, including using the next generation sequence data. In 2022, Dr. Appiah was nominated to serve on the technical panel for diagnostic tools or diagnostic protocols of the International Plant Protection Convention as a virology expert. Last year, he received training in genome editing from the University of California, Davis, UC Davis. And this was under the auspices of the African Plant Breeding Academy with the support of IITA, AUADA NEPAD, and the World Agroforestry, Bayer, Morrison Forester, and other sponsors. He's played a pivotal role in the establishment of the latest center in Gaec, and this has enabled Gaec to be able to undertake research in genome editing. Like Professor mentioned, Gaec has one of the two labs that have been certified by the National Biosafety Authority to carry out work in this area. Um, Currently, he's playing a key role in the establishment of genome editing platform here in Bnari, with a funded project in rice for the rice mottle virus resistance in some farmer prepared rice varieties. He's also a lecturer here at the Graduate School of Nuclear and Allied Sciences, and he mentors a lot of students. Our last panelist is Professor. Hold on, let me try and get my facts right. Professor Amite is one of the people I personally look up to. Came to meet him when I first came here. And he's a graduate from the University of Ghana, specifically, and he's also a graduate from the Tokyo University of Agriculture and Technology, where he did a master's in bacterial genetics. And this was under the auspices of a 2-year Japanese government Mombusho scholarship. He went on to obtain his PhD in plant molecular genetics and breeding at the Heidelberg University in Germany through the International Max Planck Research School PhD scholarship. In 2003, September, he joined Bnari. That makes him one of the oldest in Bnari currently. And he's risen through the ranks to become a principal research scientist and an associate professor. His research focuses on the application of genetic engineering and genome editing for enhancing crops, and he's been trained and has gained expertise in these areas, mainly from the University of Missouri and the University of California, Davis. He's primarily interested in utilizing genome editing techniques to create better crops with enhanced climate-resistant traits. Being an active research scientist, he has numerous publications in peer-reviewed journals. At the moment, he's the head of department at the Department of Nuclear Nuclear Agriculture and Radiation Processing at the School of Nuclear and Allied Sciences of the University of Ghana. And he's been a faculty member since 2007. He teaches and supervises students in their masters and PhDs. He's finally an IAEA expert and has conducted several expert missions where he provides lectures and training to scientists and technologists across Africa. Ladies and gentlemen, Professor Samuel Amite, our third distinguished expert for today. Right. So, just in time, it's exactly 10 o'clock, and our online participants are waiting to hear. What is genome editing? Um, for those of us who have followed the conversation around DNA, biotechnology, and the whole hula baloo, you'll be wondering, what is genome editing? What is different about it? And we have a very good expert who's going to take us through the science of it. And he's going to take us through the principles of genome editing. He's going to take us through the principles of genome editing and how it is applied. Ladies and gentlemen, once again, let's welcome Professor Valentine into it.
morning. So thank you for having me. Actually, we are always fighting each other, Nigeria and Ghana, especially when we are talking about jollof and football. So at the moment, I don't know who is always victorious, but I think Nigeria. Okay, so thank you for having me. Actually, it's the third time I'm coming to Accra. The first time I came to Accra, I went to Waki, and it was also on this aspect of genome editing. There was a workshop that was organized by Alliance for Science, and I was invited to give a talk. I was there. And then that was last year in June. And then in November, I was also here where I met the director of Bnari. So actually this is now becoming my second home because I'm always coming here. One of the Ghanaians I knew who is seated here is Professor Samuel. We met in the US in 2015 or thereabout, in 2015. We were invited by the Gates Foundation for a workshop. We met them. And since then I've been working with Ghanaians. So as the director said, my coming here is actually sponsored by AOCC, that is African Crop Consortium, and African Plant Breeding Academy. So they're actually the one that trained Andrew and Samuel. And I was one of the trainers. So here they are my students. I can tell them to do whatever thing. So they are my students. So I actually trained them. I was one of the trainers. And eventually, I was the only African in that group that trained them. The rest were from the US. And we also had an Indian that actually she's working with IITA. I actually worked under her, Dr. Lina Tripathi. I think many of you know her very well. She's a sound scientist in the area of genetic engineering and genome editing. So I give all kudos to her because I trained under her, and that's how I got the expert knowledge of genome editing. So this morning, I will be talking about the science of genome editing principles and applications. I think it should come. Is it clear? You people can see very well? Because it seems it is dull. Ah, the camera. Okay, okay. So we have been hearing about genome editing, and at times, as the moderator said, people will be asking what is the difference between genome editing and genetic engineering. Of course, there are a lot of differences, which I will also show here. So, please, the next slide. So, as an outline of my presentation, I will first of all look at the definition and tools of genome editing. Genome editing using CRISPR, because many of us came to know about genome editing because of CRISPR. Actually, genome editing has been there a few years back, before even the development of CRISPR. But it is the development of CRISPR that actually made genome editing to be well known everywhere, because it is a simple tool that almost everybody, every lab can easily apply. Before that, there were other genome editing tools like zinc finger nuclease and talent and meganucleases that people were using. But because they were a little bit difficult to apply, so it did not go around very well. But with the development of CRISPR, many people now started knowing that there is also a part of genetic engineering that is known as genome editing. So we'll talk about that. Now we'll talk about the differences between genome editing and GMOs, because this is actually a bond of contention in all our countries, in Europe, in Africa, in Asia. Whatever you do, they think that, oh, you are just developing GM. And of course, GM is not bad, they say. It's just that people don't know exactly. They feel that, oh, GM. So I will talk about that. Then we'll talk about application of genome editing in agriculture. Here, I will emphasize on the crop we have worked with Dr. Lina Tripathi, that is banana, because she works on banana. So I will give various examples in banana. Then I will again give some examples in animal for those of us here that are animal scientists. Then I will talk about the regulatory structure of genome editing in Africa. And I am happy that Ghana has developed the guidelines. And of course, as the director said, Bnari and Waki are the only institutions in Ghana that have been given the approval to do genome editing. And of course, 2 of the projects are already running in Bnari. So we'll talk about that. Then I'll also talk about capacity building on genome editing. This is actually what I trained Andrew and Samuel. That is organized by the African Orphan Crop Consortium and African Plant Breeding Academy. The training is done 6 weeks in a is done every year. The aim is to train as many as about 50 to 80 African scientists on genome editing so that they can arm the continent with people that can do genome editing. So I'll talk about that. Next. So now we go into what genome editing is all about. So if they ask us what is genome editing, so you can see the definition there. Targeted and precise changes in the genome, the genetic makeup of an organism, resulting to gene modification. So what this is, you just go to the organism and look for genes that you can modify. Genes that you can modify is either you either decide to make it get high expression or knock it out so that the gene will not have any functions again. So you don't pick genes from outside and bring into the organism, but rather you are dealing with the genes that are present in the organism. You try to modify that genes by either knocking them out in some cases, and that is one of, in fact, that is even the most application, or you try to elevate them or silence them without bringing any foreign gene from outside, unlike the GM where you have to bring genes and introgress. So that genome editing is done by nucleases. Nucleases are molecular scissors that cut the DNA, producing double-stranded. DNA is double-stranded. Don't worry about it. I think I'm having cough because I have been having it. So even if I drink that bottle, I will still be coughing. Sorry about that. So it produces double-stranded break. Why are we saying double-stranded break? Is because the DNA is double-stranded, and so when the scissors or nucleases cut the DNA, it makes a break. So the 2 halves, the DNA becomes into 2 halves. 1 half goes to the left, and 1 half goes to the right. But we know that DNA can never remain like that in the organism. So what the nucleases do is they cut the DNA, but the DNA has to repair itself. And so what happens during that repair mechanism? So there are processes in the cell that actually help the DNA to repair itself. So the cell has what we call non-homologous end joining that actually brings back the DNA, the half of the DNA together, or homology-directed repair that will also try to bring back the half of the DNA together. So it is in the course of that repair mechanism, by bringing back those half DNA together, that is where you have the mutation occurring. So that is when some of the nucleotides, because DNA is made out of nucleotides, some of the nucleotides can be deleted. A few of the nucleotides can be copied somewhere from the same DNA and put, or you can also see a base pair replacement, substitution. So that is what happens in genome editing. So from the explanation I've given, you realize that we are not taking genes from anywhere. It is just the genes in the organism itself that is being modified. But for it to be modified, we need a scissors to cut that gene. And that scissors is what we call nucleases. And there are a couple of those scissors that are used to cut the gene. We shall see them later. So, as I have explained, that resulting to index, deletion, addition, and substitutions of nucleotides. And when that happens, the gene is disrupted. Now, I want to give a practical example. I don't know if it is a suitable one. All of us probably have bicycles, or one time in our life, we have got bicycle, maybe you were on the bicycle riding and so on, right? Now, there is a chain in the bicycle that makes the bicycle to run. You have the bicycle chain. Let's consider that that bicycle chain is the DNA, okay? So, you see the chain has the upper and the lower level strand, okay? Now, let's consider that that is the DNA. If that chain is in a perfect order, you will be able to pedal the bicycle. If that chain is too long, you will not be able to pedal the bicycle, right? And if it is too short, it will not be possible. So now let's take that chain. If you cut it into two and remove some components and join it back, by the time you put it into that bicycle, what happens? It will not fit, right? You will not be able to ride the bicycle. So, but the bicycle is still intact. Nothing has happened to it. What has happened to it is only that chain, that DNA that you have got. Now suppose that chain is the one that is helping the bicycle to develop diseases. If that chain is helping the bicycle to develop diseases, like in the case of rice that Andrew is working on, the rice yellow mottle virus, if that chain is the one that is helping, by the time you put that chain and the virus attacks what rice, it will not have any way to go. It will not be able to develop diseases because you have cut that chain. It doesn't fit very well, right? Now, if you replace the bicycle chain, one of the compartments, with a chain, with a compartment that is coming from a motorcycle chain, and put it there, it will not fit. You will not also be able to ride that bicycle. If, again, you decide, when you cut it, you pick other bicycle chain compartments and join there that are longer than the usual chain that was removed from there, what happens? It will be too long. So that is exactly a scenario that genome editing is all about. You cut, in the course of the repair mechanism, a lot of things happen that it is that you make the gene to be too short, but in this case, because genes are made up of nucleotides, genes are made up of nucleotides, amino acids, and so there is always that shift in frame that actually results in the gene not functioning. So that is just a practical example. So when you cut the gene, the bicycle is still intact. If you remove a gene, if you mutate a gene from the body of a plant, the plant is still intact. But the only thing that you have disrupted is that particular gene that probably is helping something not to work well, or you are removing it so that something is going to work well, which are the typical example. Have you have gotten the science now? Yes. So the mutations, and that is why many regulatory bodies are not classifying genome editing not under GMO, because they feel the mutation that is happening species, same mutation that is happening in nature, or exactly the mutation that you can create using radiation or using chemical mutagenesis. That's why they have now regulations or guidelines in many countries that don't classify some products, not all, because some products of genome editing can still have some genes incorporated, but not all. So they have developed those guidelines that if a product does not have any foreign gene, it should not be regulated as GMO. Okay? So because the mutations are comparable to mutations that are happening in nature. But however, not all the tools that have been developed that will create double standard break. Yes, because it's not all the tools that will have the scissors that will cut the DNA into two, okay, creating that break. But there are some that you can actually program them to target a specific gene and make that gene to increase its expression, or target a specific gene and make that gene to reduce its expression, depending on what the gene is doing in the organism. And those tools are known as CRISPR activation or DCAS. So we are talking about Cas, we shall see later. Next slide, please. So several tools or nucleases have been developed to actually perform the function of gene editing. And one of these is zinc finger nucleases. Another one is talent. And then the almighty that has made all of us to know about genome editing is CRISPR. So talent, zinc finger nuclease, and CRISPR, all of them create double-stranded breaks. But the difference with them is that in zinc finger nuclease and talent, they use a different enzyme that will enable them to create the double-stranded break, which is a FOC1 enzyme. But their mechanism, no, their production is a bit challenging compared to CRISPR. So the cluster regularly interface short palindromic repeat, and the associated protein is actually very simple, easy to use, and you only need a little molecular biology background to be able to develop this. And since the development of CRISPR, many laboratories that did not even know about genome editing are now using CRISPR to develop genome editing products in the laboratory. We shall see what CRISPR is all about later. Next slide. So now I will emphasize on CRISPR because that is what Sam and Andrew are using here in Ghana. So I will take a little more time to talk about CRISPR and all the work we did with Lina Tripathi in IIT in Nairobi. We worked with CRISPR. So naturally occurring defense mechanism found in a wide range of bacteria. One DNA sequence will be repeated over and over again with unique sequences in between the repeats. So what the developers of CRISPR were studying bacteria. And what they realized was that they saw DNA sequences that were chopped into pieces and clustering the bacteria. And so they were wondering, what are these things? And why are they chopped into pieces? So when they studied further, they realized that the bacteria was chopping these DNA sequences and clustering around them to protect them from invading viruses. So that was a kind of their own defense mechanism so that viruses will not be able to attack them. And so when they studied further, they saw that most of these short pieces were just about 20 to 40 nucleotides long. And then they were always repeated in a palindromic order where the left looks the same, the left to right, and the right to left read the same, a palindrome. Most of us that have done biology will probably know that. So left to right and right to left will read the same. And so they looked at it and realized that this short DNA are actually like a palindrome running the same from left to right and right to left, and the bacteria was actually using this to protect themselves from being invaded by viruses. And so the actual thing in the bacteria that was chopping this viral DNA is the Cas. CRISPR-associated protein is the Cas that was actually cutting them. And so based on that, they developed the Cas protein. And now they have different Cas. So what they brought first was the Cas9. They name it Cas9. Cas-associated protein 9, which is the scissors that actually cut the DNA. And so, next slide. So, in this case, the Cas actually does not cut the DNA, but what the Cas does is that when you fuse another enzyme to the Cas, it renders the scissors useless. Rather, instead of cutting the DNA, it helps to activate the expression of the DNA. So that's why it is called decast. You fuse it with an activator, a transcriptional activator. And once the transcriptional activator is fused to the Cas, it helps the Cas now loses its nucleus activity and then helps now to express, to induce the expression of the gene. So this is CRISPR activation. So these are the types of CRISPR tools. Next slide. There's another tool called base editing. So CRISPR has actually developed to several areas. So in the case of base editing, you only go and flip one particular nucleotide. You don't touch any other thing in the DNA. So depending on this uses a deaminase. So depending on the deaminase that you use, you can flip, change a C to a T in a particular segment. This is guided by what we call a guide RNA. We shall come to that later on. So once you introduce your guide RNA with this base editing mechanism, it goes to the area where you have the guide RNA and flips and changes a C to a T or an A to a G depending on the deaminase you use. So it's only one nucleotide. You see how it has evolved from cutting the DNA and joining to just changing only just one particular nucleotide in a particular position without any other thing being touched. So that is base editing. The next one, prime editing. Yeah. So this is another type of editing which we call prime editing. In this case, you don't create double-stranded break. It is more programmed, but you can, what happens here, you don't change one nucleotide. You can change as many nucleotides as possible, but what happens, you need a particular enzyme called reverse transcriptase enzyme that is fused to the, like we have here, you have a pointer, the guide RNA, okay, up there. And then cut one strand, but not the double strand, and the repair of that strand will not change the nucleotides. That will result in actually changing a few or more nucleotides in the system. But the problem with this is that it is a bit difficult to manage. You have to be more expert in the area before you can manage this one. The one that is easy is the CRISPR-Cas9 that all of us are using. The next slide. So, the differences between genetic engineering or GMO and genome editing. So here, we have to look at this very well, because this is where all our regulators and so on, the anti-GM, that are now bundling genome editing under the same, like GMO, that's where they are. So we have to take this if at all those people that are outside hearing me. So for genetic engineering, you get genes from external sources. You get genes from external sources. So if you look at this, let's say that is a foreign gene. And you have identified that gene and seen that that gene is actually very good. For example, a gene that is comparing resistance, or like, let's say like the BT gene. You identify it. And then you isolate the gene. You isolate it and insert it into the organism of choice. And that organism will inherit that foreign gene and continue with it throughout life. Okay? So that's how genetic engineering or GMO is done. But then in the case of genome editing, as I explained earlier, now this is the gene. This is the gene that you have identified to modify. What you do is you design one short fragment that is called the guide RNA. So what the guide RNA does is that it directs the scissors, the nuclease, the Cas9 nuclease, to attach to a particular area. So the guide RNA is designed from the gene itself and not from any other gene. So if you identify a gene that I want to target this gene and mutate it, you identify, you design a guide RNA of about 20 nucleotides from that gene, from a particular position. Because we are talking about targeted mutation. Unlike EMS that is random, but this one is targeted. So you identify, let's say for example, this is the gene that you want to target. You identify this region and design your guide. This is the guide that you design from this region, from this gene, from that particular species you are targeting. And then what you do is you fuse this with the Cas, with the nuclease, and then deliver into the cell. So what happens is this guide now binds directly to that same position that it was designed from the gene. Binds there. And once it binds there, the nuclease recognizes this region. Recognizes that region. And then makes a break exactly in this region. Okay? Makes a break in this region because of the presence of this man here that you have removed from the plant or from the animal, and you are putting it back to the animal to help the scissors to recognize the area so that it will be able to cut at that particular position. So the scissors recognizes this area and cut here, and then that repair mechanism, the cell repair mechanism comes in now and asks, why did you cut me? I have to repair myself. So in the case of repairing itself, nucleotides are moved. This is long, okay? And then by the time you get here, it's short. Because when it is repairing itself, some of the nucleotides have been removed. You have repaired the gene, and it is now short. So that is exactly the difference between GMO and genome editing. And that is why most regulators are saying with genome editing in some countries, Ghana is one of them, this should be treated like conventional mutation because you are actually dealing with the same gene. Next slide. With genome editing, so foreign DNA free products, because that is where, and this is an important area that we should look at. Can we produce a modified organism that does not contain any foreign gene? With GMO, practically almost impossible. But with genome editing, yes. Yes. There are several methods. In the case of plants, because we are using, depending on the delivery method, if you are using the plasmid delivery method, of course, that plasmid will contain, I know that all of us here are scientists, if we have done biology. The plasmid will contain some markers, promoters, terminators, and other genes that will be delivered into the plant. But what happens is that you can cross them and segregate out. And the final product will be free of any transgene. And that one now is the one that goes out as a transgene-free product. And that is why the regulators are saying, if you have this, you can take it as any other thing to the field without passing through the regulatory as GMO. But by the time you get the final product, you cannot just release a product. So you are meet with the appropriate body, but not the Biosafety Authority, because at this level, it is now transgene free. That's one aspect. Another aspect is that you might not do it by plasmid delivery. What you can do is RNPs. RNPs means you only get the guide RNA and you fuse it with the protein and then just deliver into the cells. So the editing occurs within 3 days. And once that editing occurs, the guide RNA, which comes as primers, and the Cas9 are degraded by the cell protease. Just like the COVID vaccine. I don't know how many of us took COVID vaccine. When COVID vaccine came, people thought that it's going to integrate into the system. But this is an RNA vaccine. I don't know how RNA integrates. It's only DNA. Yeah. So what COVID vaccine does is that the RNA vaccine, when you inject it, it goes into the cytoplasm and activates the antibody to act against what? The virus. And in the end, what happens? The vaccine itself is degraded. Yeah, exactly. That's what happens here. So by the time you put this into the plant, it activates, it causes the mutation, and the cell degrades all what you have put there. Because you are putting it, you are not putting it in any plasmid that will integrate into the system. So it just the protein and the guide, it just mix them together and deliver into the cell, and they cause mutation and it is degraded. So that's one. But you cannot have this in genome, in a GMO. And this is one of the most important aspects that the regulators have considered. For you to see that they have, designed, or if you don't have this, it shouldn't be regulated as GM. Next slide, please. So, regulatory aspect. I will take a little time to emphasize it. Regulatory aspect. I was talking about non-homologous end joining and homology-directed repair. So, in genetic engineering or in GM, all final products are regulated as GMO. Okay? But in the case of genome editing, what happens is final products may or may not be regulated as GMO, depending on the pathway. But because we don't want to go through the stress of getting people to regulate this thing, to approve this, to fight with the farmers, to fight with the anti-people, to fight with those that even have no idea on genome editing, people now take the part of producing this. SDN1, site-directed nuclease 1. Mediated by this, no foreign gene inserted do not give rise to novel genetic elements in the final product not regulated as GM. So, here, these are the different tools. Okay? If you go through this pathway, with the repair mechanism, non-homologous end joining, you are going to have those indels. If you have a product of this nature and your final product does not contain a foreign gene, it is not regulated as GM. And most researchers and scientists are following this pathway. And that is the pathway that Samuel and Andrew are following, because they don't want the rice to be regulated as GM. So the final product that they are going to get will not contain any transgene. So they are using the site-directed nuclease 1 mediated by non-homologous end joining repair mechanism. But there is another pathway too you can use. If you use a pathway that is mediated by homology-directed repair with or without a template. So in the case of homology-directed repair, you will need a template. So you can get a template from another organism and instruct the Cas machinery to copy that template. So when it copies that template will remain in the organism. So now this becomes like almost like GM, because you have actually picked somewhere and asked the repair mechanism to copy that template. many scientists don't want to go through this pathway because we have seen what is happening with GM, regulatory aspects, and so on. So most scientists are taking this. So you can also go through this pathway, but in the end, you might have only one small nucleotide appearing or get integrated into the final product. It will also not be regulated. So this is site-directed nuclease 2. But site-directed nuclease 3, using this pathway, is always regulated as GMO. And that's why I said it may or may not. But in most cases, especially for those that are working with sexually propagated crops, in the final end, you will not have the transgene integrated because you will segregate out by crossing. And then you are free, it will not be regulated as GM. So anybody that thinks of doing genome editing should think of using this pathway because you are, especially if you are working with sexually propagated crops that you can easily cross. In the final analysis, you will end up not having a product that has transient regulated. You will not be fighting with NBA. You will not be fighting with the farmers. You will not, in fact, farmers, you will not even fight with farmers because if you produce rice, for them, whether there is transient or not, provided it is giving them what they are looking for, they will get it. The issue now is other people outside that have little or no idea, they will start fighting you. So it is method that all of us are working on. And that is the method that Andrew and Samuel are using. So the final product of rice that will be resistant to diseases, in the case of Andrew, that will be drought resistant and soil tolerance. In the case of Samuel, will not have any transgene. And so NBA will not even regulate it as GMO. What they need to do is, once they do all the testing and see that it is okay, they just have to meet the seed body that regulates, okay, just take it there, just for approval. Okay? So this is the mechanism of getting products. But with genetic or GMO, you cannot get this. Yeah. And that's why genome editing is making the waves. Because it will actually, but it's not a silver bullet, let me just emphasize. Because there are also things that probably you might have to go through GM. Okay? So, next slide, please. Mode of delivery application, we can forget that one and go to next up. Let's go. Yeah. So, application of genome editing in agriculture. I picked agriculture because the institute that I came to visit is actually working on agriculture. And I am also working on agriculture. So that's why I picked agriculture. So with agriculture, there are a lot of things that have been done on genome editing in agriculture. You can see salinity tolerance by modulation of NA gene. You can see nutrient enrichment, flood tolerance, herbicide tolerance, heat tolerance, drought tolerance. So Samuel might want to consider these two pathways in order to produce his rice that will be resistant to salinity and drought. And then you have disease tolerance, what is it? Pathogen tolerance. There are a lot of pathways. So these are the areas that you can regulate easily. Cold tolerance, yield enhancement. So there are a lot of things. And again, as I said, with this, you don't remove genes from outside. You just have to go into the rice gene. Which gene can I target in the rice? That if I knock it out, will confer tolerance to drought or salinity or to viruses. And they have already identified a couple of genes that they want to knock out. Okay? And if you see the studies that have been done so far, rice is one of the most edited crops because it is just like a model plant. Yeah, a model crop plant. And then the next is tomato, maize, wheat, and others. Banana, cassava is here. I don't know, I cannot see from here very well. Yam is there. I mean, banana is there. But the most, imagine this pie chart is rice. So that means a lot of things have been done in rice that will enable them to actually get their job. And I think that in just probably in the next 2 or 3 years, Bnari will be releasing or will be announcing to Ghana that they have edited rice that is either resistant to virus, and that is a very big plus to them. Next slide. So in the case of plants, I'm using plant as an example because I'm a plant scientist. How do we deliver our Cas enzyme to the plant? We can use by plasmid delivery using agrobacterium, or you can do it by particle gun. Please, next slide. Next slide, let me go quickly. Okay, yeah. So by agrobacterium, this is actually the method. I don't want to go too scientific, but for those people they know, so this is a method that you can use. You just have your plant, you have your plasmid, you put it in agro, and then you regenerate the plant. So it goes through a pathway of tissue culture. Okay? So that is organogenesis, and this is somatic embryogenesis. So those are the two pathways that we can use to develop plants, either by organogenesis or by somatic embryogenesis. So this one goes through somatic embryos. This one, you just get a tissue and then regenerate. But rice, for rice, they will be using this pathway. Yeah, because you have to develop callus and then inoculate the callus through somatic embryogenesis. You get plants. When they phenotype, and then they will segregate out the plant. The next slide. Or you can do the particle gun. So particle bombardment, we are representing this as a gun. It is not a gun per se, but because of the sound it makes. So this is actually the apparatus. It's very expensive, 30,000 USD. Yeah. But this method is not because of the efficiency, many people prefer using the agrobacterium. And I don't think that you want to waste money to buy this, except the plant is highly recalcitrant to transformation with agrobacterium. You might decide to have this equipment and optimize. But other normal circumstances, because of the high efficiency and low level of transient incorporation into the plant, agrobacterium is preferable. Next slide. Yeah. So now I'm going now to the practical examples. I've just given you a little background of genome editing, the science of genome editing. Though not too detailed, because of not everybody is in the area of molecular biology, so I just gave the surface. If we were to teach in a normal class, that then we would have gone detailed, because this is where everybody will be interested. The practical example, how do we apply it? Now, I work with Dr. Linda Tripathi, a renowned scientist in IITA. She's from India. She's known all over the world for genetic engineering and genome editing. And I'm very happy that I worked with her. Very good lady. So we worked on banana. I don't know. I brought this because these are the various uses of banana. The girls these days, in those days, you can give Valentine gift in this package. Huh? You see? You can use the leaves as a packaging material. If you give this to a lady these days, you are dead. But our parents were having this because they were living in the village. Where would they see all those fancy things to give? I don't know. Do we have this here in Ghana? This is cocky. We don't have. Okay, this one is mostly in Nigeria and Cameroon. Yeah. So this is beans, but I am not saying why I brought this is because of the material that you can use to actually prepare this. This is plantain leaf. Yeah. And then, of course, we were eating this. I grew up in the village. At time when you go to the farm, you don't have plate. You can just go and cut a plantain leaf, and they put food for you there. You'll be eating, and you'll be very happy. Okay? Of course, you can package. So, of course, banana is an important crop in And Lina is one of those first scientists that worked on banana, from genetic engineering to genome editing. Okay? So now let's go and see why she decided to choose banana. One, these are all the constraints of banana production. Black sigatoka, fusarium, nematode, weevil, bacterial wheel, bunchy top, banana streak virus. So in our laboratory, or the laboratory, yeah, it's still my laboratory because I can go there anytime. With Lina, we focus on this bacterial wilt. We focus on banana streak virus, and of course, fusarium. We're also focusing on this. So, banana bacterial wilt is prominent in East Africa. Very devastating. You can see the symptoms. Banana streak virus is prominent in West Africa because it's a virus that integrates into the genome of the plant and remains there. Okay? And banana has two genomes. I hope with genome A and B. And that virus integrates into genome of plantain and remains there. So this one is mostly in East Africa. This is in West Africa. And of course, the Panama disease that is actually destroying banana everywhere. So because of this, Lina was thinking she could be of help to the banana industry and farmers by producing banana that is resistant to some of these diseases using genetic engineering and genome editing tools. Next slide. So we are into now what we did proper using genome editing. The first is for us to establish a genome editing platform in banana. And one of the genes, the first gene we targeted was the PDS, phytoin desaturase. Phytoin desaturase is a gene in the carotene biosynthetic pathway. This is PDS gene. That converts phytoin to colorless lycopene. So what happens is that when this gene is present, it converts this. And at the same time, it makes the plant to have that chlorophyll, the green pigment you are seeing. So, why did we target this gene? We targeted this gene because if you mutate the gene and your genome editing mechanism is working, the plant is going to lose chlorophyll. All the leaves that will be coming will be white. They will be albino. That is when you will know that you have successfully established genome editing platform. So that was why we decided to target this gene. And that is one of the genes you can target as a marker to actively establish genome editing in your laboratory. Next slide. So when we targeted that gene, and as I said, banana has two genomes, genome A and B. So the genes are repeated. In genome A, you have gene, and in genome B, you have the same gene. So we look into the genome sequence of banana, and we identified that the PDS gene is 2. One is in genome A, and it has 14 exons, very big. And the other one is in genome B, and has only 4 exons. So we needed to mutate both genes at the same time in order to achieve our result. Otherwise, if we mutate only in one genome and allow it in another genome, then we are going to have maybe some partial. So we needed to mutate both. So we designed our guide, because that is one of the principal things, guide RNA. So we designed our guide targeting both genomes, so that in genome B, it is found in exon 2. We targeted exon 2. The same guide in genome A is targeting exon 5. That is for 1. So we did 2 guides. Then another guide in genome A is targeting 6, and in this one is targeting 3. So we made a construct and deliver into the banana cells. And we regenerated the plant. You can see from the results there. What happened? These are the regenerated plants. What do you see? The white. All of them that are coming are white plants. that already gave us an indication that we were able to mutate that gene, so chlorophyll was no longer produced. That's why we are seeing white plants. We also had some that had a mixture of, we call them variegated. We had some that, because of chimerism, you can see some white and some are growing, and then they control plants. So we did that for two cultivars. One is Tukalindisi, which is sweet banana in East Africa, and then Gonja, which is the plantain. So we developed that platform for both banana and plantain. And when we sequence, even though we have seen that this is already mutated, we needed to confirm what type of mutation. Because you can see this, yes, but what type of mutation? And for us to confirm that mutation, we had to sequence these ones that are white, and of course the red one. And this is what we saw here. So you saw that from all of them, even if it is just one base pair, one nucleotide, it changes. You see, like this one that has one is variegated, one is complete albino, and then you see these are the ones that we had. Some you have just a few nucleotides that have been added, and some complete that have been removed. So with this system, we now had confidence that we can do genome editing. Now, at times we do these things. When we started the genome editing, nobody taught Lina, nobody taught me. We were doing it on our own. You just need molecular biology background. Okay? So we had now confidence in this, and this is, you can read that publication, you can read about this work in this publication. Next slide, please. The next thing was, could we actually go and knock out the integrated version of banana? I told you that banana streak virus integrates into the genome and remains there. Yeah, and so it is actually a major problem, because especially for the banana that has genome B, people will not want to transport it because you'll be transporting the virus. So it's almost similar to the AIDS virus, because the AIDS virus also integrates into the body and remains, right? So, but it is difficult for you to work with humans to actually mutate that AIDS virus and remove it. But here we had to do it. So what happens is that it has, the banana streak virus is in two forms: the integrated version and the episomal version. It's the one that actually integrates into the system. And when conditions become unfavorable, the integrated version reverts to episomal version and then produces the symptoms. So, and you see that this integrated version has a lot of inverted repeats of the same operating frames, because it's made out of 3 operating frames, 1, 2, and 3, and you have a lot of inverted repeats. So what we did was, could we actually knock out this, so that in the end, we will not have banana that will produce this virus. So we will not be able to revert to this, and that symptoms will not be produced. So we did our bioinformatics studies. We designed 3 guides targeting operating frame 1, 2, and of course 3. So the operating frame 3 is actually very big with a lot of genes. So we targeted those 3 operating frames and developed our banana. And then we got the plant. We did mutation, sequencing analysis. We got a lot of mutations. Here. Next slide. And then we check, because one of the things that actually activates the integrated to revert to episomal is when conditions are not favorable, like drought. So we took the mutants together with the wild type and then subjected them to drought in the greenhouse. When we did that, we saw the results. 2 of them, 3 of the mutants did not present any symptoms. I don't know if you can see clearly from there. Yeah, 3 of them, 1, 2, and 3 did not present symptoms. The first one there is the wild type. The wild type means is the control, non-edited. And then the other 2, this and this, got symptoms. And we're wondering, if they are mutated, why are they having symptoms? Why is it that out of the 5 we tested, 3 have symptoms 3 are symptom free, 2 have symptoms. Just why? So we went now into our sequencing analysis to actually check very well what happened there. And when we went into the sequencing analysis, so we now realized we came up with this model that for you to actually have that virus completely knocked out, you must have knockout in all the operating frames. That if you only knock out operating frame 1 and 2, you are going to have partial inactivation of the virus. The plant will still produce symptoms. If you knock out all the 3 operating frames, all this and this, you have virus inactivation. If it is only this, you are going to have partial inactivation. They will still produce symptoms. And so, why this and this produce symptoms is that only the 2 operating from 1 and 2 were knocked out. The last one was not knocked out, and that's why we're having partial virus inactivation, and of course, we're always producing symptoms. So, for that banana virus, people are using this method to work on HIV, but they are not doing it in experimental organisms like rats and so on. Maybe with time they will come up with a solution, because they are using this genome editing in medicine, and already they have already got success with sickle cell. Yes. So next one. So that is about. So we started with developing the platform, mutating PDS. We went into knock out what? The banana streak virus. Okay, we have done about virus. Now we are going to bacteria. So for bacteria, we edited a susceptibility gene. What are these susceptibility genes? These are genes that actually are found in the species that aid the pathogen to colonize and produce symptoms. They're actually there to aid the pathogen to colonize and produce symptoms. So if we target those susceptibility genes, the pathogen will not have anywhere to colonize. So one of those genes is, there are a couple of them. One of them is the downy mildew resistant 6. An S gene. S there is for susceptibility. So one of them is this. But in most cases, before you go to knock out a gene, you have to do a lot of research and tests. First thing is, what we did was, we inoculated the plants with the bacteria and allow it to develop symptoms. When we inoculated with the bacteria, we now check the expression of this gene. Because you can knock out a gene that will not have any effect. And that is what we were yesterday with Sam. He inoculated his plants. And he wanted to know whether in the presence of the virus, the gene that is targeting is actually going to be elevated. So when we inoculated, this is a wild type progeny. This one is resistant. Is resistant to Xanthomonas. So when we inoculated, this is the control uninoculated. This and this are the resistant wild type that is not used. You see that there is no elevation of the gene, but then the susceptible one has very high elevation. So based on that, it gave us confidence that we should target that particular gene. So we did that. Design guides, 2 guides, targeted, delivered. Next slide, please. So we sequenced, after getting our plant sequence, we saw a lot of edit, mutation. And then now we tested the plants, first in vitro, and then second in the glass house. When we did that, in the in vitro, this is the wild type. They were producing symptoms. The mutants did not show any symptoms. And when we took them to the glass house, we saw the same result that we saw in vitro test. The mutants were very healthy without symptoms, whereas the control died. So it shows that the mutation of DMR6 is an important gene that if you mutate it in a plant could confer resistance to bacteria diseases. So that is one of the summaries of the findings. And in banana, it has happened. Next one. In the same way, we saw another gene that we decided to knock out. It is called L-nodulin, N-node. Nodulin genes are involved in nodule formation in legumes. And they are formed during nodule formation. But then, some time ago, they saw that some of them are also involved, are also seen in non-leguminous plants. That shows that it is not only, they are not only involved in nodule formation, but they could also have some functions. Okay? So one of them is early nodulin. Next slide, please. So we targeted early nodulin gene. Okay, I'm going. When we targeted early nodulin gene, we got the plants and we checked the results. The plants were the wild type, the mutants were resistant. Next slide. So now we also check, could we activate instead of just knocking out? Could we activate? We produced a construct that had a transcriptional activator fused to the Cas9. In this case, we are rendering the Cas9 inactive. We don't want it to mutate. We don't want it to cut the plant. We rather want the gene to be expressed. We did that. We delivered into the plant. Next slide. And again, when we did the test, these are the mutants that had high level expression of the gene we were targeting. We're targeting 3 genes for each to elevate the expression. This also showed resistance. That shows that whether you use the knockout or you use the activation in banana, it is working. Next slide. Nutrient enrichment. I've talked about developing the platform, disease resistant, bacteria and virus. Now, this is for nutrient enrichment. You can increase the nutritional composition of your plant. This work was not done by us. This work was done by another group. I brought it here so that we could see the various areas. Nutrient enrichment. So this is again the carotene biosynthetic pathway. They did not target PTS. They targeted it. So their aim was to increase provitamin A. Okay, beta carotene accumulation. And of course, we know the function. So they targeted this so that when the lycopene will have to be under by lycopene at lycopene CB will now convert this to beta lycopene. To beta carotene. It will get accumulated. So they targeted this so that it will not go through this part. It will go through this pathway. What did they see? High accumulation of beta carotene in banana. So that is one of the things you can do. Next slide. Plant architecture. If you want a plant to be short, you can do editing. So what did they do? This is another group that works on banana. So they targeted the gibberellin gene. And when they did that, what is the result? This is the wild type. Significantly short plants they got for banana. What was their aim? Banana is usually tall. And in the rainy season, they topple. And they don't want it to be toppling. Because when it topples, you are going to lose your bunches. They don't want. So they targeted that. And they got significantly shorter plants. Next slide. You can increase the shelf life. Shelf life. Here we can work on tomato. Tomato is, especially we in West Africa with the high heat and so on. You can increase the shelf life So they targeted this gene in banana and produced plants with increased shelf life. The banana fruit up to 80 days. But look at the wild type. Just 21 days, they were already decaying. This is the mutant. Took them up to 80 days. So that is what genome editing can do for us. Next slide. So there is another genome editing tool that we applied. It is called Cas-Clover. With Cas-Clover, the Cas9 is not the one that is cutting the DNA and making double-stranded break, but rather it is Clover 51 that is doing. So we also tested this method in banana by targeting PDS. Of course, as usual, we got yellow plant by using that method. Next slide, because I don't have time. We again developed the same system for yam. So now we have worked on banana and yam. In yam, we developed using the PDS. You know, yam is very, very difficult to transform. And so one of the things we did was, could we actually develop that system using the PDS that we could easily say we did that for yam, and we, of course, we got albino yam by targeting the PDS. Next slide. Now, that is, we are done with the plants. I mostly showed you the work we did. Now, this is what other people did in animals. For those of you that are in animal, cow, hornless cow. Why did they do it? You see, they use editing to produce cow without horns. The reason is that when they have horns, they will always be fighting each other, harming each other. So they produce cow without horns using editing. And that is the gene they targeted. They targeted that gene. Now, virus. They produce chicken that is resistant to this virus by targeting this gene. And one of the most interesting is red tilapia. That was done in Israel. Why did they do it? They said the black tilapia was no longer sellable. People were tired of buying black tilapia, so they want to do yellow tilapia. And so they edited this gene. They edited this gene and produced yellow tilapia. You see what editing can do. Next slide. Mushrooms. For those of us working on mushrooms, non-browning mushrooms. That's also a kind of decaying. So they produce mushrooms that will remain completely white. They don't decay. A genome editing. Next slide. Okay, I've already discussed about this. Next slide. Next slide. I don't want to waste time. This is too scientific. Next. Okay, now. Regulatory aspect. It is not coming. Why is it not coming? No. Why is it? Yeah. Is that part? Go. The next slide. Next slide. It is not coming. Go. Okay. Let's make it came. So these are the countries that have actually developed guidelines for genome editing in Africa. Kenya, Malawi, Nigeria, Ghana. We should clap for them. They are not staying behind. You see, they don't want to be left behind. And I told you that Ghana is always competing with Nigeria. Always competing. Whatever we do, they do. Whatever they do, we also do. You see? So with this, in Ghana, you can go and get your lab certified, and then you can start your genome editing work. And that's what the director here did, and got so Sam and Andrew are in good foot. Next slide. So capacity building. When the director started his welcome address, he emphasized on African Crop Consortium and African Plant Breeding Academy. They are actually championing the training of African scientists on genome. Their aim is to train as many scientists as possible. And so far, they have trained these scientists from these different countries. Burkina Faso, 1. Ghana, 2. We are always fighting with Ghana. We have up to 6. Malawi and so on. So these are the countries that have been trained. So they are training people they don't just train anybody from anything. They train from countries that have guidelines. either JEB or guidelines, or you have molecular biology laboratories that you can do the work. Because it will not make sense for them to train you, and in the end, you don't do anything. Okay? So, I don't know if you can recognize anybody from here. Who can you recognize from there? Yeah, that is Andrew. And then here, that is Professor Amite. Yes. So they are undergoing this training. So they have done class 1 and class 2. They have just advertised for the next class. So maybe you can send in your application. They will share with you that advert. You can send in your application and get trained. Yeah, they train you and do a follow-up. So I'm actually doing the follow-up. That's why I'm here. They do a follow-up so that they will be able to establish, okay, for 1 year. So next year again, you people will see me here. I'll be coming for some. Yes. Next slide. So in Ghana, as I was saying, for now, I don't know if this is current. I don't know if the director knows. I don't know if there's anything going in Waki. I have no idea. I'm not sure. Is there anything going on? Nothing. So far, it is ordered in Bnari that the projects are going on. The PI of this one, resistant to diseases, is Andrew. And then salt and drought tolerant is Samuel. And eventually, imagine all of them are going on in the same institute. So that means they will be the first. So we have to thank the Director General. Of course, the Deputy Director is here. Yeah. So I'm thanking him. Thanking him, please, when you go, extend my regards to him. I thought he was going to be here, but I heard that he has been called by the ministry for because I went to his office. We stayed, he started, and he's actually promising to make the center of excellence for genome editing. And I'll be glad. I'll be glad to help in whatever way. And if Rita hears that, Rita is the one coordinating this African plant breeding and the training. She'll be very happy, and she will actually do everything to support. Next slide. Well, this is a take-home message. Just we want to encourage all of us, Africa, to actually adopt genome editing. It's very, very good. The most important thing is that you can end up producing a product that does not have any foreign gene. It will just go out as conventional breeding. That's the most. And then the training. So Sam and Andrew, and of course my director here, try as much as possible to train as many people as possible, because we need those hands. Yeah, we don't just need one person to. In fact, we are not living in a society whereby I want to be the king alone. No. Yes, as many people as possible. And if people that are not even Africans can go look for money, they look for money to train this. It's very expensive to train one person, and even to pay us, those of us that are training them. If they can do that, why not we? So please, try. I've seen your lab. It's okay. It's good enough. In fact, the tissue culture lab is the one I like best. Excellent. It has the facilities. The molecular biology lab, I said probably because you rush and put it on, try to put a few items. There are a few items that are not there. Who was writing this today as we were talking? So he wrote a few items. Let's work with the director to put it on. I tell you, genome editing is one of those things that is going to drive the world, even in terms of medicine. I did not put medical things here because I did not want to put something I'm not best, but I know that it has been done in sickle cell. It has already been delivered. So you will see. So next slide. Now, these are the people that have met us to really know about genome editing. And they won the Nobel Prize in Chemistry in 2020. This lady spoke, that is Jennifer Carpentier. This one was not recognized, but actually it's one of those. There's no black person there. We should have a black person. You can develop a product. One of the advantages is that you develop that product, you patent. You become a very strong person in genome editing. Next slide. So, this, I am in UM6P, University of Bamesik Polytechnic. It's one, I would tell you, is now one of the best universities in Morocco. If somebody told me that I will ever go to Morocco, I would have asked him, are you mad? You know, the negative thing is, when I was in Saudi, I did my postdoc in Saudi 3 years in King Abdullah University of Science and Technology. If you see that school, you will not believe that you have that type of thing in Middle East. Then one of Sam's classmates who came from Morocco was the one who even encouraged me. Come to Morocco. I said, my friend, are you stupid? What am I coming to do? Come, my friend, you will enjoy the place. So fortunately, they advertised a position and I applied. I went there and I just opened my mouth and said, hey, do we have this type of thing in Africa? If you see the facilities, the working conditions, ah man, those guys have gone far. And this year, I advertised for positions. I've already employed 3 PhD, one from from Nigeria, from Morocco, 2 postdocs, one postdoc, one from Pakistan. So, and each year, they advertise a lot of PhD positions. Those PhD positions are fully sponsored. You don't spend fishy. Yes. So, take note. This year, the application probably is closed. Next year, once they advertise, I will alert Sam and Andrew to advertise for you people, for those who want to do PhD. Just get experience. It's not a matter of, if you don't, this one is just to help you not spend money from your pocket, because they are going to pay for everything. Whoever I've employed, I'm going to pay for everything. They will be there. So make sure you apply next year. Once they advertise for those PhD positions, advertise. When they advertise, apply. Because you might be lucky. They do a lot of, they have a lot of areas. Not only on editing, they have a lot of varieties of areas. We just had interview last time where we interviewed somebody from Ghana, and I think he might get a position in there. So be checking. So most of those work, you heard me calling Lina. She is actually the principal scientist of in the area of biotech in IITA. There's no other person doing it. I work with. And then this is the Plant Breeding Academy, UC Davis. Those are the main sponsors, together with African Open Crop. I brought in these people and this because some of the work in banana was sponsored by them. Next slide. Thank you so much. Sorry I took so much time for trying to explain a lot of things.
talents. And then to the almighty CRISPR system, specifically the one that Jennifer Charpentier developed at 9. Then we looked at the differences between genome editing and genetic engineering. And he also took us through regulations and made us aware that different countries have different regulatory frameworks for GED or genome editing. Some regulate it just like they do with GMOs. So in Africa, when you show the map, you saw South Africa was red because South Africa sees everything as GMO. In Ghana, Nigeria, Kenya, and Malawi, they have 3 categories. And those 3 categories, he mentioned SDN1, SDN2, SDN3. There's a very simple way to remember SDN1, SDN2, SDN3. If you get a cut on your hand, there are 3 ways you can solve it. You can leave the cut for it to heal, SDN1. There's no foreign assistance. SDN2, you can put a plaster on it. That helps you to heal, but it doesn't form part of the final one. So you can take the plaster off and your body will heal on its own. SDN3, you can have a skin graft to cover it up. It means you brought something foreign to cover it up. He took us through the applications, and there are so many applications of it. And we were privy to what Lina and her team at IITA, which Prof was part of, have done. So this is good for us when it comes to yield, improvement, drought. Our crops in the north currently, it's sad. The report we get from the north is very sad, where especially for maize, it has started, forming the cobs, and there's no rain. And even if it rains now, they've lost. For disease, and I think it took us through a lot of the diseases. Plant architecture, maybe one day those of us who are short will end up getting tall, like the plant, or those of us who are too tall can end up getting short, maybe. And I'm happy work on banana and shelf life has been looked at. Then he took us through the crops that are being worked on globally. Rice is getting a lot of work because now you cannot go a week without eating rice. But rice has a lot of issues. So we need to work on these crops to make it more available for us. Tomato is also following rice. I think last month or 2 months ago, China announced huge amounts of money that will be putting into wheat research. So those who are against this technology but will be importing wheat from China in the next few years will be having it on their tables. He spent some time talking about banana, about the work on virus, and then on the bacterium, and on nutrient enrichment. Having pro-vitamin A banana is one that we should encourage. And the beautiful one is in animals. When I first heard about the hornless cow, I asked myself, why would you want a hornless cow? But when you realize the issues that come with cows having the horn, then you understand why there's a need for hornless cows. Some countries around the world have already approved some animals for eating. Canada, I think they've all done genome edited organisms that are on the plate of the people. So that was a very insightful lecture. We are going to the second part where we have a panel discussion. There are a few questions that we want to ask them. And so, with your kind permission, can I invite our experts to the front? I have some quick questions, 3 quick questions, and then I'll open the floor for all of us to be able to ask. Yeah. So, thank you very much for availing yourself. Yes, I'll sit. I usually like to put them under stress, like the teacher who is invigilating. Okay. My first question, I think a lot of people would want to understand: is Africa ready for this science? After all you said, do you think we are ready for this science? I don't know who will take it first. Maybe let me start with Prof, and then I'll come.
Thank you for that question. Actually, Africa is very much ready for the science. If you look in Africa, you can see that one of the things that will make you know that Africa is ready because some countries are already developing. They have developed guidelines, and some are developing. That is one. 2, African countries, AUD NEPAD is doing a lot of communication on genome editing to actually make people aware of what genome editing is. So that is one of the things that you will see that Africa is ready. 3, Africa also has facilities. I went to the University of Ghana yesterday, Department of Biochemistry. They have everything it takes to do genome editing if they decide in the center of excellence, if they decide to do it. WACI has everything. The Atomic Commission has everything. And that is not only here. If you also go to other countries, like even where I am, we have everything. And besides, we also have experts. You don't really need to be expert in genome editing to start genome editing. No, you only need to know molecular biology and have the facility. So in a nutshell, to me, Africa is ready, very ready to adopt this technology. This technology is not like nuclear science that people think. What I have presented here, if you see, is as simple as that. It doesn't take so much. Very simple to do it. So many people were thinking that you will need big equipment, you will need maybe, I don't know, you have to be a witch or wizard to do this thing. No, it's not like that. It's not like that. It's very simple. You can do it. You only need those basic things. So Africa is very much ready. The only thing we need to do is to encourage the government to actually adopt the technology, because many governments, they are still having that issue of GM, it is GM, it is not GM, it is GM. So if most of these countries can develop guidelines to specify how it should be done, then I don't think we are going to have any problem. Thank you.
Let me ask Dr. Pierre. There's a worry of potential off-target effects. What mechanism can be put in place to forestall any off-target effects that may come from doing genome editing?
Okay, thank you very much, Dr. Fosu. When the speaker was talking, he made mention of guide RNAs. Okay, the guide RNAs actually will lead the molecular scissors to positions where they bind and the cutting is done. Now, during the development of these guide RNAs, there is what we call off-target analysis. So that is done to make sure that the guide RNA that have been designed will bind to the exact locations where we want to do the cutting. So when that is done, you minimize the occurrence of off-target. Off-target means that the guide RNA is binding to sites other than where you want it to bind, and then you have all sort of phenotypes, that is expressions in the organism that you do not want. So these are all taken care of, and during the process we do it in a containment. So whatever is not good will not come out. Thank you.
Thank you. So my last question will come to Prof. Can you spend a few minutes to tell us your work, what exactly it's on? Prof mentioned it, but can you give us an idea on how Ghana is potentially going to benefit from this?
Thank you very much. As it came out clearly, this is a program that we are running, and it's for the first class, and I'm doing the second class. It happened that his project that he's working on is a biotic trait, virus resistance. So the one challenge with genome editing is most of the time researchers will do either the biotic traits or abiotic traits. And research has shown that it's normally quite easy or easier to do with the biotic traits. So being a new lab going to start, the idea was to target the easier traits at the beginning. Then once we are quite mature, we can then delve into other areas. So it came to a point, how then do I design my project or research? Then the temptation is to try to also go for a biotic trait like he's doing. I said no. Yes, there's that challenge, there's that difficulty in editing research, but somebody will have to go for the abiotic as well. And abiotic means you are looking at traits like drought, you are looking at traits like salt tolerance, flood resistance, etc. These are more what I refer to as climate resilience traits. So I decided to pick on salt tolerance and drought tolerance. Approach my supervisors how to defend it, and they say, well, somebody will have to champion that. So go ahead, let's see what comes out of it. The good thing is these traits have been tried in this country before. That was the newest rice project. And so newest rice was looking at nitrogen use efficiency, water use, exactly. So, but then that project had some challenges. I talked with the PI involved, and he gave me a background to it. So for this particular research where I'm going to look at drought and salt tolerance, I'm taking a particular lesson from that project. And there's something I plan correcting so that this project does not hit the sort of challenge that one had. Thank you.
All right, thank you very much. Let's give them a round of applause. And I find the work on the drought tolerance or the water use efficiency and salt tolerance very interesting in rice. A lot of the rice we grow in Ghana is upland rice. So we grow them in lowland areas, and so it needs a lot of water. I'm sure all of us know that our farmers are growing in areas that are marshy. But with these drought conditions we are having in most of the growing areas, it's possible that you would lose your crops. Now, the other thing about rice or about crops is that when they are drawing water from the soil, that water is not just water, it's bound to salts. And so these salts would rise with the water and they get left on the surface of the soil. So if you realize that the soil that you're using to grow your rice becomes a bit more salty. And when you grow it, if you go to those fields, rather than seeing green, um, you see it yellowing, and so you will not have enough grains come out. So it's a beautiful one that I think Ghana will benefit from. Our rice farmers will benefit from. I see a huge audience here, so let me have a round of 3 questions, and then if you have burning questions, yeah. Yeah, so let me see 3 hands, and then we'll 1, 2, 3. Okay. Let me start with you.
Yes, so thank you very much for a wonderful presentation. Just one quick question. I would like to find out from you the message you have for our farmers who are suffering, who want to have high yield crop, especially rice and other things. What message do you have for them so that they will have faith that something new is coming to them and they will buy into it? Thank you very much.
I also want to find out the guide RNA. The guide RNA, is this specifically designed to be inserted into the plant or something, the cell, or is something that you go and buy and then put it in? I just don't get it very well. So please kindly help me on that.
You're saying is that do they have to design? Do they design it, or they go and buy? Okay. So I saw a third hand here.
Thank you very much. Hello. Thank you very much for the presentation. Please, my question is, after the guide RNA binds to the part of the DNA that is supposed to break and the cutting is done, how do you ensure that the cell repair process has the nucleotide that you want to have the mutation occurring?
So let's take these 3 questions, and then we'll go back again. For our audience online, please, you can type your question, and I'll ask it to our expert.
Yes, I think the first question was what message we have for our farmers. And I think the message will be one of assuring them of the biosafety protocol that will go into developing these products. And then the other assurance will be that the products that will come out are going to be products that will benefit them economically in agricultural production. Now, the reason I'm saying for the biosafety, they should be assured is that we have the legal framework in place. And as the presenter mentioned, that will lead to the development of this product. But then our target is only to go through the pathway, just developing the mutant, which is the SDN1. So we are not going to venture into the other pathway. So biosafety, we are clear. It's cleared. And so we believe it is going to be a landmark process pathway that will lead to etc. Thank you.
Well, let me add to that. Let me add to that. No, a farmer is always one of the things the farmer wants is to get seeds that he will be planting. Seeds that will be coming year in, year out. So I know that there was an issue with GMO that farmers were people were thinking that the seeds that they produce or that they use, you can only produce for 1 year. The next year you will not be able to get. But this is not the case. Yeah. So farmers should be assured that once they get this seed from either rice seeds or what, they'll be able to be multiplying and using. So in that case, they will increase their productivity and make income as he has mentioned. Because one of that's a major thing for farmer. Can I have seed? Will I be able to get money in this seed? So farmers will not spend money on buying pesticide, insecticide, or whatever thing. Okay, because you're having seeds that are resistant to particular diseases. So that money that they would have used to buy this pesticide, insecticide, they will put it in their pocket. So that's it. So the second question he asks about the way designed.
I also want to add something to the case of the farmers. What I want farmers to do is to allay their fears on these modern technologies. Now, at first, people were of the view that these technologies are being done somewhere and they are coming to be imposed on us just to cause us not to have enough seeds to plant or to increase the or possibly to kill us. But then now the technology has come to us. We are the ones going to catch to do the production. And believe me, I will not produce something that is harmful that will also take part in eating. That will be equivalent to suicide. So scientists will make sure that the right is done because we are also going to eat the products of the genome editing that we are producing. So have faith in us, have confidence in us, and trust that we are always going to do the right thing and bring the right products to you. Thank you.
Yeah, I like that point because that is one of the things that we usually face. Most of us in Africa feel that this thing is done in the white man country to come and kill us here. Yeah, but that is not the case. The work I've shown here done in banana was done by us. I am involved. The rice that is going to be produced here will be produced by one of you. So he will not produce something that will kill you. So it's not something that will be produced in America to come and kill us. No, please erase that. So is this one message is not just for farmers, it's also for all of us. So the second question is, he asked whether we buy the guide from somewhere. I'll answer that question. See, the guide is designed by the researcher himself. But when he designs the guide, he designed the guides as primers. Yes, the guides, he designs them as primers. So he will order it as primers to be able to use it and put into the plant. So those are two things. One, he will design it by himself. And then the second part is for him to now order it as primers. Yeah. So, because if you design the guide, you are designing it in computer. You cannot pick it just on soft copy and put into the plant. It has to come as in a form of DNA because primers are just DNA. So he will now order it from any company that is synthesizing primers. And so the company will synthesize that guide as primers and give to him. Then he will now do all the mixtures and then put into the plant. So I don't know if it is all clear. Sorry.
Once they order the primers, then they will design what they have, and then they.
No. You just what the like for him, he will design the guide by himself. He will only order it as primer. Inkaba or whichever company will just produce only that one, the 20 nucleotides, and give to him. Yes, he will now bring in other things and put with that guide together and put to the plant. So the person that is producing the guides as primers has no other thing to do for you. It's only to give you as primers. Because when you design primers, they will give you as primers. They will not do any other thing. Yeah. So the guide is always designed. But when you design, we call it guide because it is something that is directing the scissors to attach somewhere. But when you are producing it as primers. Okay, so that's why we call it guide, or at times we call it, when you design it is guide, but when you are bringing it from a company, it becomes oligos. Yeah.
All right, thank you very much. The third question was repair after the cut. How would the cell know the right nucleotide and pick it? Is that right? Good. Now, when you was talking, you made mention of three repair pathways, and the first one was the non-homologous end joining. Now, this is a natural thing. It happens when the DNA are broken within cells. So when the double strand DNA molecule is cut, now the cell wants to repair itself. Now, in the process of the repair, an error is created. In fact, the non-homologous end joining is an error-prone process. It is prone to errors. And that is what we are after. And that will give us the mutation. So it is not about picking the right nucleotide. For instance, if the thing is T, and there is a cut and there's a repair and T is picked again, then of course you are not going to have any change. But the idea is to disrupt the gene. Now, when you go to biology, you have nucleotides, the genetic code, which are the triplets of bases. So ATG gives one amino acid, TTG gives an amino acid, CCG gives one amino acid. So what happens is that when there is a cut and ATG is taken off and T is put there has been a mutation. Now, when the mutation occurs, it means that the actual function of the gene is disrupted. So if, for instance, if the gene is supposed to make the plant tall, okay, and you want a plant that is short, you cut the gene that is responsible for the tallness, you remove a nucleotide from it, or you add a nucleotide to it, and that is the mutation. Now, when that mutation is done, the protein that is produced will not be the actual one. There's what we call frame shifting. Okay, so the protein that will be produced will be a frame shift protein. So it is no longer going to serve that purpose of making the plant tall. So the plant becomes short. And that is genome editing. Thank you.
If you want to ask a question, please catch my eye. 1, 2, 3. We have someone from the regulator here, so let me start with him.
Thank you so much for the presentation. I want to touch on the RISE study. I'm very happy you indicated you're already in touch with the PI for the CSIR Kumasi project. And then to encourage you that liaise with him and then for best practices so that we don't get to where he got to and then have to more or less drop the project behind. There are a whole lot of things to it, and then that is encouragement that I just want to give. That lays with him so that you know that you're on track, and then you continue with the work. Thank you.
He's talking more about collaboration.
Yeah, thank you.
I have a few questions here. So, okay, I'll ask one, only one. From the perspective of food safety, we know that there are certain compounds that are produced by most of these plants that we feed on or use as food. Now, my question is, for instance, tannins, you have oxalates, you have goitrin. Now, these are compounds that are produced during the growth cycle of the plant before finally you harvest. Now, has there been any work, or are you people thinking about any work that can, deal with some of these things? Since most of these compounds either inhibit nutrient absorption or tend to have certain effects on humans. For instance, where you have goitrin, some people have slight allergy, they are somewhat allergic to goitrin. So because of that, they are unable to feed on typical examples like cabbage, cruciferous. So the question is, are you people thinking about food safety so that individuals that are allergic to some of these compounds can, get their safe foods and consume without any problem? Thank you.
Thank you. Yes. So third question, and then we come back to you.
Thank you very much. Please, I want to know, apart from infrastructure limitations, what are some of the problems that a scientist who is working on developing a gene edited crop must envisage? Thank you.
So we have three diverse. So one was a comment or an admonishment.
Yes. The regulator has come up with a very good suggestion, and I think I mentioned that I'm already in talks with the PI, and he's giving me some pointers which I've captured, and I'm going to take note of that as I roll out this particular research in rice. The other question was on food safety. From the presentation that we had, now genome editing is quite a versatile technology, and so basically your question hovers around quality product development. And so if it's an issue of tannins, basically simply like Prof described, you go through the catalog of genes that have been implicated to control high tannin production. And then you, through genome editing, knock out some of these genes. And then you have products with low tannin or whatever. Just as they were able to raise the beta carotene levels, you can always go through, knock out whatever you don't want, and you are good to go. So it's not really, right now we are not thinking about that. We are beginning, so we are starting with few traits. But with time, we'll move into food safety, food quality, things like post-harvest, etc. All these fields are there to explore. So let's just relax. We'll get there for the food scientists too.
Okay, so that question, Professor Sam, thank you. You have answered already that question. So he will take the one on problems so that I will not be the only one talking because I already gave a presentation. But I also want to add to what he has said. You see, when anything you are doing, you do something that you are trying to address a problem, a major problem. You don't just start. If food safety, like the tannins and so on, we know it is a problem. If it is not a very big problem per se, you might not have to consider that for now. So you have to start somewhere. And the somewhere they are starting is what they have identified to be an issue. And as he has said, with time, people will also pick those ones up and work on them. But addressing a major problem is one of the major things that we have to look into. Yeah. So those ones will definitely come when they would have trained a lot of people. And then that's when they will now spreading their tentacles. Well, look at this. This could be a challenge. This could be a challenge. But so far at the moment that is not a major challenge that is giving people a major problem. It might not be a priority for now, but it might be a priority later on. So that's what, because they cannot just go doing things at the same time. They have to start gradually until they get there. Yeah. So some people, like for example, I developed a project that, okay, I should work on taro, cocoyam. You know, auxiliary cocoyam. It itches very well. Okay? Unless you cook. There are some that even when you peel it, your hands are in danger. Okay? So I'm thinking of developing that project, but unless we have people that will be able to maybe employ and work on them, then that project will also be. That's one. Two, another challenge of actually working on, at time for them that are sponsored, For them to develop that project, they also have to go in line with what the sponsors want. So that's one of the things. And some of the crops that have these problems are probably not priority crops for some of the donors, because you need money to do some of these things. So you will not just go do it if it is not a priority crop, because some of these funding bodies, they have their crops that they want to fund. So that's why maybe some of these projects are geared towards those areas, because you also need money. You don't want to start something that in the end, you remain in the middle, you cannot go forward, you cannot go backward because there's no money. Yeah, thank you.
All right, thank you. I think the question is about problems in executing genome editing project, right? Apart from infrastructure. Yeah, at first, I'll talk about training need. But currently, with the help of AOCC and the UC Davis, AUD, and NEPAD, a training program has been rolled out to train scientists in Africa for genome editing, and I'm a beneficiary. Professor Amita is also a beneficiary. So for training, I can say for now, some training. So we have it, but the major thing I will talk about is funding. You may have the training, you may have the infrastructure, the equipment, but we need funding to execute some of these projects. So I'll use this opportunity to call on the government and other funding agencies to support research in this direction, because Ghana has a huge problem when it comes to agriculture. Climate change, the effects of diseases, and all that. So we all need to work together. I mean, we have institutional support, we have training, we have infrastructure. So what is left will be the funding. I think that will be the major problem as of now. Thank you.
Yes, so what I want to add, yes, training looks quite okay now, but we have to continue with a lot of training, because the two of us you see here will not be in this business forever. So that will. The other thing which I want to point to, which those who will be assisting us in the laboratory should take note of, is that this is a very fast evolving field, technology. Almost every day something new is happening. And so if you don't read or follow up and track developments in this field, it will get to a point you appear obsolete. You won't be able to cope. So that is the other challenge. As a researcher in this field, you must always be reading and learning. Thank you.
Thank you very much. Can we give them a round of applause again? I know if I'm to ask the floor for questions, we will have a lot more, but this is the start of a conversation. This technology is here. Okay. We have some farmers here. Yes, I see a hand. Let's hear from one farmer.
Thank you very much. Please, I will speak our local language so that Mr. will be an interpreter for me. Thank you. Pachawo challenges be a phone be yenya. Ye mobile na ye piano. The bit is a cabbage, tomatoes, and Jenny. Ye Jenny seed na ye piano e be fi Nigeria. Na nyami huni say omo Jenny ni ba ebo akesiye. Ebi soso pa na me hu say sa bana na ye so ye den egu ha na ye dua. But at the end of the day no ye dey need to any omo dey any compete. Ebi hwe na dey na ye nkete. Akesiye ni wo mu ye kakra bi. Ye mu nti a eba ne sa. Na to so no ye cabbage. Udua cabbage na say every day another biara na ji obadru. at the end of the day okra no kyerɛ sɛ ɛbɛsɛ yi wobɛkɔ back wobɛbɔ duro several occasions but at the end of the day wobɛbɔ ka but cabbage no bi tumi ba ma wohu a aboa biara kora ntumi fa n'ano nkae the same as yɛ tomatoes mpakyɛw pii ehyia a yɛhyia yɛ mmoa bɛn na yɛn mpanyinfoɔ yi a ɔmo frɛ yɛn ba yɛ ɔmo bɛtumi de aboa yɛn ɛfa gyeene n'aba a yɛbɛdua na yɛ akɛseɛ ne mmoa a ɛyɛ a ɔmo hwere yɛ cabbage a at the end of the day na cabbage no bɛworɔworɔ ho ara na kaketeketekete nti mpakyɛw bi nso ne question no nti with the message ma wakyerɛ sɛ kakra ma.
Thank you very much. So the question is a very simple one. Cabbage faces a lot of challenges when they grow it. You end up virtually losing the cabbage to insects, particularly the diamond back moth. And so he wants to know what can be done to help them when it comes to cabbage. On the other side, we also have onions. He knows they get their seeds from Niger, Nigeria, and other places. But the onions that come from there are quite huge. When they plant those same seeds here, they end up getting very small onions, and the large ones are very few. And the same problem they have with tomatoes. So what solutions can you prefer for them?
Sɛ woka twi no me nso mede twi no bɛfra kakra sɛde bɛyɛ a asɛm a mede ma wo no nso wobɛte aseɛ wo sɛ deɛ ɛdi kan no mo seeds no monya firi burkina sɛ niger good ɔmo dua wɔ hɔ a ɛbɔ akɛseɛ akɛseɛ ba mode na mo dua a na ayɛ nketenkete nketewa ne dɔɔso ta akɛseɛ no nti deɛ ɛdi kan a I will touch on is the conditions a ɛwɔ Ghana any conditions ɛwɔ baabi a mokɔfa seeds no ɛnyɛ pɛ nti sɛ as plant breeders when we come out with a new variety we have what we call multi-location trials nti multi-location trial ne sɛ ma yɛ gene ɛyɛ new variety gene ne bɛkɔ abɔnten yɛbɛdua wɔ Bunahafo ne yɛdua wɔ Greater Accra Kudu Ashanti ne yɛdua bi wɔ Central ne yɛdua bi wɔ Volta ne yɛhu sɛ ɛbea bɛn na saa gene no yɛdua a ɛbɛyɛ yie paa na yɛde ahyɛ report to na aba sɛ because ebi a ɛhɔ no gene bi a nsuo ɛtɔ hɔ no different from aha sɛdeɛ mo dua no no the agronomic practices na moyɛ wɔ ha no ebi a ɛnyɛ the same thing ɛna wɔn nso yɛ wɔ hɔ ebi tuma ba they are doing something better than you are doing here okay nti ne nyinaa ka ho bi ɛna ɛma gene no akɛseɛ ne nketewa no ɛnyɛ sɛ odua adeɛ no nko a sɛdeɛ wɔbɛtete no the way you nurture it the agronomic practices that you do and the prevailing environmental conditions ne nyinaa ka ho bi ɛna ɛma gene no ɛbɔ wate ase nti nyɛ sɛ bi a deɛ ɔmode ma mo no na ɛnya anaasɛ biribi nti ɛno no wobɛtake note of it yeah the cabbage first
cabbage mmoa no yɛte aseɛ now that same problem no yɛhu wɔ cowpea mu ana beans that problem no na ɛwɔ beans mu but yɛkasa sesei no yɛtumi use technology wei a develop beans a sɛ mmoa kɔ ho a wɔn ntumi nsɛe no ɛna ɛyɛ sɛ quite recently na Ghana release saa nti saa cowpea no ɛnkyɛ obi a odua cowpea bi abɛnya bi na odua a wode compare na ɛwɔ hɔ dada wobɛhu sɛ mmoa no wɔn ntumi wei yɛmmɔ mpaeɛ sɛ onyame bɛhyira yɛn na yɛnya sika bebree yɛbɛba cowpea cabbage yɛbɛ the same technology wɔ cabbage nti sɛ yɛdevelop saa cabbage no a wobɛhu sɛ a problem yɛ a mokɔ mu ɛsibɛtwa that's it.
Thank you very much. So what I want to add quickly, let me speak to you and then I'll say it in English. In Central for mo ne ɔmo nkasa na mo nka mo haw nkyerɛ ɔmo na ɔmo fa mo haw no ɛnto dwa na ɔmo a ɔmo nim kuayɔ haw a ɛwɔ mu no ɛnte aseɛ ɛsiane sɛ sɛ wodua cabbage a wobɔ spray ɛnɛ na nsuo tɔ a walusu wobɔ spray ɛnɛ ɔtena akyi ɔsɛ ɔkɔbɔ spray wambɔ a mmoa no ɛwe nti wotumi bɔ spray a wobɔɔyɛ ana wɔɔtɔn mɛboa aha nkurɔfoɔ bebree nte kuayɔ ase ɛna ɔmo nte mmoa a abɔdeɛ mu nyansapɛfoɔ yɛde boa kuayɔ nti nsɛmtwerɛfoɔ wɔ ha ansa wɔbɛkɔ no wo ne ɔmo nkasa na ɔmo mfa w'ano kasa no ɛnto dwa na amanfoɔ ɛnte aseɛ sɛ sɛ anim anyɛ adwuma ambɔ mu a kuayɔ no ɔhaw no yɛntumi ɛnsoa no ɛkɔ so saa a wobɛgyae na wakɔyɛ bi foforɔ. So what I just said was that we have the media present, and so it is good for them to carry their message to the media so that those who don't really understand the problems that farmers face with their farming practices would understand that there's a need for scientists to work together with these farmers for solutions. Without these solutions, agriculture is gone, it's lost. And farmers, because they have to spray, if they don't spray, they lose. With time, they get tired of it and they'll move on from agriculture into other sectors of the economy, and agriculture becomes unattractive to other people to get into.
See, that is why when we are developing any project, we develop a project in collaboration with the breeders and farmers. The breeders actually understand what the farmers want. We are in the field of genome, let's say in the field of biotech, probably will not, especially if we are in the university. In the university, professors just want to develop something, get their papers, they don't even think of products. But if you are dealing with breeders have pretty much idea, because the breeders want something to go out and not just I get something, get publication, that is it. So breeders work with farmers. So the moderator said, so there should be that close collaboration between the breeders and the farmers, so that the breeders will understand what the farmers want, what they are facing, and then they will be able to work with scientists that are probably in that area that will be able to bring out solutions. When that is done, farmers will face less challenges, and they'll be able to have some of the problems solved.
A huge round of applause for our experts. We're way past our time, and there's a need for us to close. They are going to be here. If you have any further questions, you can ask them. Prof, okay, so Prof wants a last word. And then
as a head of department, I'm always advocating for students from my department. And so at the Department of Nuclear Agriculture and Radiation Processing, our doors are very open for any students out there across the country who want to be trained or acquire your degrees in this area of research. You are welcome to join our team. Thank you.
Thank you very much.
Right. Something short for our farmers. Please, all that we are saying here is that there is a new technology that has come. Technology biaba, your friend genome editing. Now genome editing, no, it me and yama bebre. Inti ano ho nkomo na yedi na ye hyase o binary ha ye de emo na starting. Ye de emo na hyase. Na sa genome editing no, itimi ma planter emo awe no itimi ye na emo awe biom. De embo kese no itimi de ye na abo kese. De ye pese dan no ho kakra ye titia itimi ye na ye titia. Eno ho nkomo na ye di. Enye ade bia etire so. for genome editing. So corner be obi busa wo say asama ye kan no ase ne de na wo de ba fia technology bi aba e fe ne genome editing. Scientist na sha se o Ghana na no itimi boa ma yin aya e be boa yin na e se be bo se yare enchi no. Okay, thank you.
All right. Okay, so I want to call on Professor Adumako to give us his closing remarks. It's been a very long morning. Yeah. So
we are already in the afternoon. Hello. We are already in the afternoon. So I believe you all agree with me that the presentation given by Professor Valentine Intui was very educative and insightful. Prof, I'm a physical scientist, so all these things, most of them I don't even know. But I have been able to enjoy your presentation. I've taken a lot back home, and I'll begin to be contacting them to learn more. The one interesting thing has to do with the genetic engineering and the genome editing. There's a vast difference between the two, and I think the scientists will be able to take it up and propagate the information out there. Yeah, and as for the advantages, they are enormous, and I believe, because my first time of hearing that this hornless cow came through genome editing is very interesting, and mushroom production and a whole lot. And one thing that I want to emphasize here has to do with communication, because as you are doing this thing at the lab and the farmers are here, me being a professor, I don't even understand what you are saying, how much more then. So it will become a very difficult thing. So how do you tackle it down? So communication should be a key in this regard. You develop it, how do they get it, and how do they also propagate the information out there? Possibly you have to meet the farmer associations, and then you break it down. You break it down for them to understand the technology that you are embarking on and the benefits that they are going to derive from it. It's very important. And the other thing that I'm also very happy has to do with the succession plan. Succession plan is very important. Now we have only two. Human beings are like our vehicles that we use. Today you drive home safely. The following morning you go, you sit in the car fully prepared, try to put the engine on, says no, I'm not moving. That's how human beings we are also like. So succession plan is very important. So you have to try as much as possible, get people involved so that people will be able to take it up when you are not there. And the other one has to do with collaboration. I like the way the gentleman said, what are you doing with CSIR? You know, the most research institutions that we have in Ghana is CSIR and Ghana Atomic Energy Commission. And you have to work hand in hand. There was a project about 10 years ago. I was on it with CSIR. There was a Canadian fund to that project, and they were using the conventional methods alone. So when the Canadians came to me, and I said, I'm going to use both conventional and nuclear technique. So I am adding something to it. And they asked me, what kind of nuclear technique are you going to add to what the CSIR people are doing? I said, I will be able to tell you the age of the underground water. And they said, wow. And I told them that I will be able to use nuclear technique to tell you when it rains, how much water goes to add into the underground water system. So because of that, I got the project. So if you add conventional to nuclear technique, it edges you up. And so some of these things, collaboration is very important. And we should also try as much as possible and develop confidence in whatever we are doing. Because as you are going out there, people will be asking you a lot of questions. So you should be able to articulate yourself and let them know that what you are bringing on board is going to have a lot of benefit for humankind. On this note, I thank you very much, and I wish we are taking a lot back home. Thank you.
All right. I think on the program you will see that I have to give the vote of thanks. So thank you all for coming. Well, we want to say a very big thank you to Almighty God first for making this happen. And then to thank Professor Adumapo for gracing the occasion, giving the welcome and then the closing remarks for us. Thank you very much, Prof. We thank our keynote speaker. We caught him at short notice, but he's demonstrated that he is a true professor. He's always prepared to deliver, and he has delivered excellently on the topic. Thank you very much, Prof, for accepting to speak at this webinar. We thank our panelists, Dr. Andrew Sakode-Appiah and then Professor Samuel Amite. They are our trailblazers when it comes to genome editing in Ghana, and we look forward to seeing greater things coming from their laboratories. We thank our partners who are here. We invited our colleagues from CSIR, from NBA. I can see the NBA people are here. We invited colleagues from the university and other research institutes. And then we have our students who are on internship. Some are doing national service in our laboratory. Students, can you just wave us? Yes. And then our beloved farmers. We work closely with these farmers. They are farming on our land, and we work closely with them. If they have challenges, they bring the challenges to us. So the challenge that our brother brought, cabbage, no, there was solution, ma cabbage. Into any more bekasa. Na ya share ni ayebe. Onion no send ya kan no. Ya bibi on cassava. Sure, see a bit to me replicate the same thing with the onion. They are improving a yield. Yeah, cassava there no. Now farmers no 20 tons per hectare. By a technology no they call no. Want to mean 70 tons per hectare. And see any more bekasa. Na ya share send ya bit to me abuamo. There be a demonstration. Ama yin na ya share. If then we can adopt those technologies. Into ye be ye da must pass him aba. Every higher corner. Ye also I editing komo. Na ya share send ya bit to me abuamo more. We thank all of you for coming. Our online participants, thank you very much for joining us online, and all of you in the room for being part of this very insightful webinar. Thank you very much, and have a good day.
Before the closing prayer, if you missed any part of this, it's going to be online. In fact, it was streamed live online, so you can always go back to it. It's on YouTube, and so you can always go back and relook at what was presented today. Thank you very much. Closing prayer.
Okay, shall we have the closing prayer? Shall we pray? أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين. In the name of Allah, most gracious, ever merciful, Ya Allah, we are thankful for this day. We are thankful for the gifts of life and our health. We are very thankful for a successful presentation. It is our prayer that as we depart from here to our various destinations, you will be with us and guide us to apply the knowledge we have acquired here in our various research to the benefits of mankind and to the glorification of your holy name. Amen.
Thank you all. Yes, so